So now let us do cell death. So cell death is essentially a topic that finds a mention at two places in our syllabus. So one mention it finds in developmental biology, where it is combined with aging. So aging plus cell death. And then it is also mentioned as a part of genetics, where simply it is mentioned as cell death. So as a student, how should you approach the topic? First and foremost, because it is a topic mentioned in genetic syllabus, so you need to know its molecular basis so that you can explain the mechanism of cell death properly. And when it comes to doing justice to the topic from the angle of developmental biology, then what is the importance of cell death in normal animal development is what you should be adding to your coverage. So if in one coverage, which is what we are going to do today, if in one coverage you can include both the molecular mechanism and what is the significance of the process in animal development, then we would be catering to the needs of cell death topic from both genetics as well as developmental biology perspective. You really do not need to do the same topic all over again in two different parts of the syllabus. That is sheer wastage of time and totally unnecessary, because 90% of the things will anyway be overlapping. So how are we going to organize this topic? First and foremost, we are going to understand what is cell death. Okay. So first is what is cell death? After that, what are the two major types of cell death? And let me tell you right in the beginning, one type of cell death is cell death by design, which is totally in the control of the organism. This we know as programmed cellular death. And the programmed cellular death also has a name, that is apoptosis. Second P is always silent. And apoptosis is a word that you use for programmed cellular death in animals. And the second type of cell death is cell death due to an injury. You call it necrosis. Necrosis so far has not been asked, but in the end, I am going to include a comparative table between apoptosis and necrosis. After that, we need to know the basic molecular mechanism of apoptosis. Once we have understood that, then we are going to see the significance of apoptosis, both from the angle of defense of human body and also from the angle of proper developmental requirements of not only human body, for that matter, development of any chordate animal that we know of. So under these four headings, we have to do cell death. And then, of course, the handout that I'm going to share, about 8-page handout, that will cover everything that we are going to study in this class. And ultimately, you will have enough content to write in your examination as well. So now let's start. Let's first answer the question, what is cell death? So how do you define death to begin with? How do you define death? So death is when any organism, let's say that we are talking about death of an organism. So you define death, that death is that state of being at which the organism loses its physiological and reproductive vitality. Means the organism has lost both its vitality. With physiological vitality, the organism maintains itself, organism repairs the damaged part, organism grows, carries out various metabolic processes, so on and so forth. Reproductive vitality ensures that the organism is capable of begetting more organisms of its own type. So when an organism loses both physiological vitality as well as the reproductive vitality, and if the organism also loses its structural integrity, and all of these irreversibly, we will call this stage undoubtedly death. The same three things, when you apply it to the level of a cell, you will be calling it cell death. So how to define cell death? Cell death you can define by, give me a moment, cell death you can define by calling it irreversible, okay? Irreversible loss of a cell's structural integrity, physiological vitality, and reproductive vitality, okay? And not to forget that these losses have to be irreversible. Means once these losses have happened, then it is not going to recover from it. That is cell death. So once a cell dies, it has been lost from the body permanently. So you can also say that it leads to permanent loss of a cell from the body. So that is what cell death is. Now that we have understood what is cell death, then we need to talk about the two types of cell death. There are two types of cell death. One is called programmed cellular death. And second is called injury caused cell death. This injury called cell death is known by the proper name of necrosis. Let's imagine a cell is suddenly exposed to fire. So the cell will immediately burn and die. Cell is exposed to a strong acid, cell is exposed to a strong base, a cell is exposed to very strong electric current, cell is exposed to dry ice. Immediately at that location, the cell will be dying. This is necrosis. Sometimes the necrotic death need not be that dramatic. Cell is constantly getting exposed to harsh chemical and it may die a slow death, but make no mistake, the cell has still died because of injury either to its structure or to its metabolism, and that is how a cell has died. In both the cases, the cell has died because of an injury, that is necrosis. Okay, but when the cell's death is actually a part of body's larger design, when the cell's death is actually a part of body's larger design, then the body controls which cell to die, when to die, and by what process should it die. This kind of death is known as programmed cellular death. In short, you can call it PCD. For all of you, you should be knowing one thing very clearly, that PCD is the broader term and apoptosis is a type of PCD. All the PCD need not be apoptosis. But in animals, the kind of PCD that you see is apoptosis. So apoptosis is a PCD with some special features. Okay. So apoptosis, and mind it, the second P is silent. Apoptosis is animal PCD and it is accompanied by certain special features. And to be precise, there are four special features which we are talking about. What are those four special features that we are talking about? Let's go back to our slide. Okay. So I hope we are back to our slide. Yes. So we are back to our slide. Now see, here, these are the four things which will always happen in a cell that is going through apoptosis. So what is the first thing that you see in a cell that is going through apoptosis? The first thing a cell that is going through apoptosis is that formation and pinching off of vesicles. And there is a name for that, that is blebbing. That means a cell that is dying will be forming outbound vesicles like this. So a cell will be dying by forming these outbound vesicles. Inside those vesicles, there are different cellular components, and these cellular components will then be eaten away by the neighborhood macrophages. Okay, so macrophages will be there. Macrophages are actually present in every single tissue of the body, most of the tissues at least. So these macrophages will be eating them away. These structures, which are outbound vesicles, they carry the debris of a dying cell. So first thing is blebbing. These structures are called blebs. This is a bleb. Okay. Second is the nucleus and chromatin goes through condensation. So initially the nucleus will be normal, but very soon the nucleus will get very condensed structure like this. Dark and condensed structure. So if this is the nucleus, over a period of time, nucleus will go through condensation. Next thing is that the DNA will be cleaved into characteristic 180 base pair fragment. So DNA, which is there inside of a cell, so this DNA will be cut off in tiny little segments. And you know what? These segments are of nearly fixed size, and the size happens to be around 180 base pair long. I would agree that it varies from one species to another. There is no doubt about it. But that is the size in human beings. And the fourth thing that you will be seeing, that these apoptotic bodies will be formed, right? These apoptotic bodies will be formed. So a bleb, when it is released, it is called apoptotic body. So as long as it is attached to the mother cell, it will be called a bleb. And when it gets removed, it will be called an apoptotic body. So it forms apoptotic bodies which are engulfed and degraded by the neighboring cells. So a PCD with these four features, a PCD with these four features is known as apoptosis. So apoptosis in all cases, right, is not synonymous to PCD. It is a type of PCD. In the plants also, there is programmed cellular death. In the plants, programmed cellular death does happen. But the plant PCD does not go through apoptotic pathway. The plant PCD goes through the death process, but not by shrinking of the nucleus, not by forming of the outside bleb, because in the plants, the cell wall will be constraining the formation of the outside bleb. So in the plants, basically the enzymes present in the vacuole will be breaking down the internal cellular content, but the plants would not be showing the apoptotic bodies, blebs, and all that. So animal cell PCD is apoptosis. I hope you have understood this much. Now, take a look on this diagram, which is going to come on your screen now. And then you see how diagrammatically you can make a distinction between these two processes. This is a normal cell if you look at it. So this is a normal cell. So when this normal cell is going through apoptosis, so it will be showing membrane blebbing, condensation of the nucleus, and then the cell will be breaking apart into several small little apoptotic bodies. And these apoptotic bodies will be engulfed by the neighborhood in the same tissue, the macrophages which are out there. And there will be no local inflammation. Apoptosis keeps happening in a lot of tissues in our body, where older cells are removed and their place is taken by new cells. This is actually a process that we know as cellular turnover or tissue turnover. So tissue turnover or cellular turnover is basically routine removal of older cells and their place is taken by newer cells. our RBCs, they die once. I mean to say, a batch of RBC will die in 9 to 10 weeks, and their place will be taken by new set of RBC. So every day in your body, 9 to 10 week old RBCs are getting eliminated, and every day in your body a lot of RBC is getting formed to take place of the RBC which are getting eliminated. Similarly, intestinal epithelial cells are routinely getting removed, some of them, and their place is being taken by new intestinal epithelial cell. So this is something that keeps going on. This is something that keeps going on. On the other hand, if you now see necrosis. So in necrosis, there is some injury, cell will swell up and cell will burst. So plasma membrane will rupture, cellular and nuclear material will simply release away from here, and this is likely to cause local inflammation. There will be some amount of inflammation. So seen that way, apoptosis is a nice and clean process. If you see here, so in here you see that a normal cell, it goes through chromatin condensation and everything. Membrane blebs are getting formed, and after that there will be cellular fragmentation. And these fragments released from the cell are phagocytosized mostly by macrophages. Macrophages, you know that they are resident of most of the tissues, so they will be identifying them and they will be eating them away, and thus a cell disappears without leaving any trace. So you have understood what is apoptosis, what is the larger word PCD, and how apoptosis and necrosis differ in their scope. You have also learned what are the 4 characteristic features of apoptosis. We are now turning our attention to apoptosis control. How is apoptosis controlled? Okay. So when we talk about control of apoptosis, first and foremost, let's talk about what triggers apoptosis. So apoptosis can be triggered by internal factors and apoptosis can be triggered by external factors. When apoptosis is triggered by external factors, mostly it is triggered by certain signaling substances. Okay, so there are certain signals which we also know as death signals. So these death signals are nothing but special types of proteins, and they are signaling agents. So these proteins will be acting upon certain receptors. 2 very, very well-known signaling agents which are associated with apoptosis happen to be FAS-L, and second is tumor necrosis factor. Right. So FAS-L is a protein which will diffuse to the target cell, and then it will be binding. No, FAS-L is a signaling agent that is present in the plasma membrane of a signaling cell, and it will be going and it will be binding with the target cell. On the other hand, tumor necrosis factor is a soluble protein. It will float in the tissue fluid, and then it will be finding the target cell and it will be binding the receptor of target cell. So when we talk about FAS-L, so this is the cell that will cause another cell to die. So this is a signaling cell, and this signaling cell will be targeting a cell. Wait a moment. This signaling cell will be targeting a cell which has got FAS receptor cells. This is the target cell. But the target cell won't die until it has been signaled to die. So what happens that when this cell, target cell, is no longer needed, so this signaling cell will go and will interact with the receptor of this one. Can you see that? That it is interacting with the receptor of this one. And now you can see almost perfect interaction. And that is how this target cell will be stimulated to die. On the other hand, if you look at the TNF, so TNF is a diffusible signaling agent, and the cells which have got the receptor for TNF, and if they happen to be exposed to TNF, then those cells will be dying. So that is external factor. So when apoptosis is controlled externally, we call extrinsic apoptosis. So when external factors, give me a moment, let me just clear it up a little. So when external factors control it, then we call it extrinsic apoptosis. Okay. And when internal factors control it, we will of course be calling it intrinsic apoptosis. What kind of factors cause intrinsic apoptosis? To cause intrinsic apoptosis, it is possible that the cell has accumulated a lot of DNA damage due to different mutagens, so on, and now the cell is unable to repair those damages. In that case, the cell will have no option but to go through death. Similarly, the cell might have accumulated some radiation-related damages to various macromolecules and all, the cell will die. If the cell is now losing its survival factors, there are specific proteins which keep the cellular processes going on. So if the cell is lacking those survival factors, cell will be pushed towards death. So there are a whole lot of intrinsic factors also which will be causing the death of the cell. In either case, whether the cell is dying intrinsically or the cell is dying extrinsically, in either case, there is a certain group of proteases which will be activated. So be it intrinsic or be it extrinsic, they both lead to a pathway. They both lead to a pathway that activates caspase proteins. What are caspases? I'm going to explain it in a moment. And caspase proteins lead to cell death. So let's first deal with caspases, and then we are going to see how caspases are activated by intrinsic pathway and how caspases are going to be activated by extrinsic pathway. So caspases are basically cysteine aspartate protease. So to begin with, it's a protease. It breaks proteins. Aspartate is an amino acid. Aspartate is located at the target site of protein breakdown. Where the protein has to be broken down, that is where aspartate is going to be present. And the amino acid cysteine is going to be present, just add E that I forgot. So amino acid cysteine is present at the active site of the enzyme. Okay, so the active site of the enzyme contains cysteine and the cutting site contains aspartic acid. Hence, it is called cysteine aspartate protease. These proteases, they cut a whole lot of cellular target proteins, and in a sequential manner, they will be causing the death of the cell. But these proteins themselves need to be activated by slight cutting. These proteins themselves need to be activated by slight cutting. So let's say that if this is a caspase, if this is the caspase enzyme, so it itself needs some cutting to be an active enzyme. Do you see there is some cutting? So this structure is called procaspase and this is called caspase. All right. And any guess who will cut a procaspase into caspase? Another caspase. So this cut by another caspase. So some caspase will cut a procaspase into active caspase. And then an active caspase will go and cleave means cut, target proteins of the cells. That within the cell there are target proteins of the cell. So within the cell there are target proteins and those target proteins will be cleaved off. Now this diagram gives you insight into two types of caspases. So essentially speaking, there are two types of caspases. The first set of caspase that we are talking about, the first set of caspase is nothing but those caspases which activate, one sec please, those caspases which activate other caspases, right? So these caspases which activate other caspases, these caspases do not target the cellular protein. They are basically activating those which will cut the cellular protein. So these caspases are called initiator caspases. What will we call them? We call them initiator caspase. Correct? And the caspases which cleave the cellular target proteins and thereby bring cell death, those caspases are called executioner caspases. These caspases are going to be called executioner caspases. So how does the process now look like? The process looks like either there will be an intrinsic stimulus or internal or intrinsic stimulus, or there can be an external or extrinsic stimulus. Correct? So the stimulus could be either. So this will be working via signal receptors, and signal receptors will activate the adapters. Adapters are associated with the receptors. So if this is the receptor, generally these receptors are dimeric, means two subunits. So with this, there are adapters associated. So this is adapter, this is receptor. And once the receptor has bound with signaling agent like this, the receptor will be active, and if the receptor is active, adapter is active. Then an active adapter will convert into procaspase initiator. Pro C, I stand for initiator, into caspase initiator. Means the job of converting initiator procaspase into caspase is the job of adapter, which has been signaled by a death signal which bound to the receptor. And the receptor activated the adapter, adapter converted the procaspase into proper caspase. And this proper caspase then converts whom? Procaspase executioner into caspase executioner. it converts pro-caspase executioner into caspase executioner. And we all know that caspase executioner will then break down cellular proteins. And these cellular proteins, when they are broken down in a sequential designed manner, then that will be causing cell death. So that is how it is progressing. Now let's talk about, we turn our attention to internal one. So mostly the intrinsic stimuli, they lead to release of cytochrome C. Cytochrome C is a protein found in mitochondria, and it is a protein that is associated with aerobic respiration. So release of cytochrome C from mitochondria into the cytosol. And once it is released from mitochondria to cytosol, this cytochrome C will convert procaspase initiator into caspase initiator, and this will be converting procaspase executioner into caspase executioner, and this caspase executioner will eventually break down cellular proteins, and that will cause cell death. So whether the process is triggered extrinsically or intrinsically, either way, the process will involve the activation of procaspases initiator, and once they are activated into caspases initiator, then they will be activating the caspase executioner. The executioner caspases will then cleave off the cellular components and will cause cell death. So that is the process. There is a diagram that I am going to show you. In this diagram, there are more pathways which are shown, but that we will come to later. Those pathways are actually not important for the examination, but those pathways also contribute to cell death. So take a look on that diagram. Here, at least in the form of a flowchart, you should be drawing the figure. Okay, so here we are. Take a look. There is a figure that is going to come on your screen in less than a second. Yeah, here we are, the diagram, with the diagram. Take a look here. First and foremost, you have got the death receptor pathway, extrinsic pathway. So the receptor and ligand is the signaling agent. They bind the two types of FAS and TNF. Then adapter proteins will be attached, caspases will be activated. So we now know which caspase will be activated. First and foremost, initiator, then executioner. Once the caspases are activated, then endonuclease activation will take place, breakdown of cytoskeleton will take place, and then can you see the apoptotic bodies are being formed? They will be released and they will be engulfed by the phagocytic cells and all that. Right? Now see the mitochondrial intrinsic pathway. So in mitochondrial intrinsic pathway, you see that there can be growth factor withdrawal here. Then there can be DNA damage by radiation, toxin, free radicals and all. There can be lot of protein misfolding and all. They can all lead to some amount of stress on the cell. Mitochondria releases cytochrome and certain other pro-apoptotic proteins. So cytochrome C is the main pro-apoptotic protein, and what is it leading to? Caspase activation. And then that will be leading to various proteins being broken down. Nucleus breakdown will cause nuclear fragmentation. Cytoskeleton breakdown will cause the formation of the blebs and all. And like I was saying, that it is showing certain additional things which you won't be needing in the exam. But if you want to put it in the exam, you are fairly allowed to put that in the exam. That if the cell has got BCL2, BCL-XL protein and all, that will what? That will block the apoptosis pathway. So these are the proteins which help the cell survive. But if these BCL-XL2 proteins are not there, the cell will be dying. So the cancer cells which avoid the cell death, they normally have a very high level of expression of these anti-apoptotic protein. They are regulators of apoptosis. They inhibit apoptosis. And then there are certain other members of BCL family like BAX and BAC. They promote apoptosis. But if BCL2 and BCL-XL is there, they will block this one also, they will block this one also. If you want to mention the role of BCL, it is right here in the diagram. BCL2 and BCL-XL will inhibit the apoptotic pathway, and then BAC and BAX will be promoting the apoptotic pathway. I have removed myself from the frame so that you can take a good look at this entire diagram, right? And this diagram is a part of your handout, so you can very well use this diagram. It will need some practice, but it is not that complex a diagram which you cannot use in the examination, right? So it is from a very, very well-known, very well-respected book in medical pathology by Robbins. So this is a diagram taken from Robbins' book, and very clear and comprehensive diagram for both types of control. And one more diagram I would like you to take a look on, and that will complete apoptosis. This is about the interaction of different types of caspases. So see, apoptotic signals have come, they bind to adapter proteins, and these initiator caspases, which are numbered as caspases 8 and 9, they have these cleavage sites. So when they associate with the adapter proteins, they will be cut at the cleavage sites, and now these are active caspases. These active caspases will be activating the executioner caspases. Caspase 3, 6 and 7 are known as executioner caspases. So they are activated by cleavage, and they are now active caspases which will carry out cleavage of multiple substrate within a cell, and that way they will be bringing in apoptosis. So that's all about molecular mechanism of apoptosis. So we have understood what is apoptosis, what are the highlight features of apoptosis, what is the control of apoptosis, and how caspases, two family of caspases, initiator and executioner, how do they drive the process of cell death. Having said that, now importance of apoptosis. Some of the examples here. First and foremost, the example is in the worm Caenorhabditis elegans. So the adult has got, in Caenorhabditis elegans, the adult has got 1090 cell. But when the adult is maturing, exactly 131 cells, out of these 1090 cells die in a pre-programmed manner, and the mature adult has exactly 959 cells. Such is the precision, and each cell, these 131 cells, they die by nothing but apoptosis. Another example, in our immune system and in our nervous system, unwanted cells are removed early during development. In the nervous system, those cells are removed which are unable to form a synaptic complex, a synaptic junction, they will be eliminated. Next is the formation of proper connection between neurons. It requires that surplus cells are removed by apoptosis. Then death of interdigital tissue, right? So embryonic hand looks like this. Embryonic hand, very early during the fetal stages, it will look like this. And then it looks like this. So what has happened? Between the digits, the tissue which was present, this tissue got liquidated, so that fingers are free. And who causes this? Death of interdigital tissues during embryonic development. And during amphibian development, the tail resorption is and apoptotic process. Then there are roles in defense. For example, cells which are infected with viruses. So the cells which are infected with viruses, they are killed by cytotoxic T cells, which are known as killer T cells. Cells of the immune system. So after the immune reaction to the pathogen, once the pathogen has been dealt with, then effector cells are removed so that they no longer attack the body's own cells. So to prevent their unwanted effect, effector cells are slowly eliminated. Next is cells with DNA damage. So cells with DNA damage, they are also removed by apoptotic process, else they will multiply and they will create a huge population of cells with persistent DNA damage. And lastly, whole lot of cancer cells are eliminated in the early stages by apoptosis. And many of the chemotherapy agents, for that matter even radiation, is an attempt to kill the cancerous cells by causing damage, and these damages will then trigger apoptotic process. So these are some of the roles of apoptosis in defense. Rest of it is basically necrosis. So necrosis refers to accidental cell death, and this will include increased cytoplasmic eosinophilia. So within the cytoplasm, you will be finding a lot of affinity for eosinophil. Nucleus will randomly fragment, breakdown of plasma and organellar membrane, just it will break down, no clean process of blebbing and all of that. Formation of abundant myelin features, small little fibrous fragments will be there, and leakage and enzymatic digestion of cellular content. Some of the cellular content will leak away and some of them will be randomly digested by enzyme. Because it is accidental, injury-induced death, so there is no specific clean pattern that you will be seeing here. This is the table of difference between necrosis and apoptosis. In necrosis, cell size will enlarge. In apoptosis, cell size will reduce. In necrosis, nucleus will go through darkening, but nucleus can simply get fragmented and it will be broken into small pieces. But here, the DNA is fragmented into nucleosome size fragments. Plasma membrane here is disrupted. In this case, plasma membrane is intact and it forms an altered structure, but it is still intact. Cellular contents go through enzymatic digestion. It may leak out of the cell. Here it is intact and it may be released by apoptotic bodies. Adjacent inflammation in necrosis, yes. In apoptosis, no. Physiologic and pathologic role, it is invariably pathologic. It leads to some kind of injured tissue, some kind of unwanted outcome. And here it is often a tool of eliminating the unwanted cell, which may be pathogenic after some forms of injury like DNA damage and all. So before such cells cause damage, such cells will itself be eliminated. So that is your short, ready to use tabular summary of difference between apoptosis and necrosis. And that's all about cell death that you needed to know, both from genetics molecular biology point of view and also from the point of view of developmental biology. So that's all. And along with this, do go through the PDF handout, which will be summarizing exactly the things that I have shared with you. So that is all.